Bismillahirrahmanirrahim. Dear students, it is our lecture number 2 and chapter number 3, X-ray diffraction techniques for MS Chemistry semester 2nd. Now we are going to discuss the principles of single crystal X-ray diffraction. Before this, we have discussed the single crystal X-ray diffraction, how the diffraction pattern of X-rays for single crystal is obtained. Now here is the principles of single crystal X-ray diffraction. The fundamental principles of single crystal X-ray diffraction. Max von Laue in 1912 discovered that crystalline substances act as three-dimensional diffraction gratings for X-rays, wavelengths similar to the spacings of planes in a crystal lattice. The X-ray diffraction is now a common technique for the study of crystal structures and atomic spacing. The X-ray diffraction is based on constructive interference of monochromatic X-rays and a crystalline sample. These X-rays are generated by a cathode ray tube filtered to produce monochromatic radiation. These radiations are then collimated to concentrate and directed toward the sample. The interaction of the incident rays with the sample produces constructive interference and a diffracted ray when conditions satisfy the Bragg's law, which is n lambda equals 2d sine theta. This law relates the wavelength of electromagnetic radiation to the diffraction angle and the lattice spacing in a crystalline sample. These diffracted X-rays are then detected, processed, and counted. By changing the geometry of incident rays, the orientation of the centered crystal and the detector, all possible diffraction directions of the lattice should be attained. All diffraction methods are based on generation of X-rays in an X-ray tube. This is the phenomena and schematic diagram showing the X-ray diffraction technique. You can see from the top, it is the source of X-rays. X-rays are produced by some source which are then monochromated. The monochromatic X-ray beam is hit on a single crystal. When the X-rays hit the single crystal, it is diffracted. The diffracted beams of X-rays are then detected by a film or any detector. The crystal can be rotated or mounted to some extent in different orientations so that we can get two-dimensional images, and when we get the two-dimensional images from different sides, we will then combine them and we will get in the three-dimensional pattern of the crystal. The X-rays are directed at the sample and the diffracted rays are collected. A key component of all diffraction is the angle between the incident and diffracted rays. Powder and single crystal diffraction vary in instrumentation beyond this. What is the importance of X-ray diffraction by single crystal? Its importance is this technique is a non-destructive analytical technique, as we have discussed in our previous lecture, which provides detailed information about the internal lattice of crystalline substances, including unit cell dimensions, bond lengths, bond angles, and details of site ordering. Directly related is single crystal refinement, where the data generated from the X-ray analysis is interpreted and refined to obtain the crystal structure. We have discussed that the X-ray diffraction technique is very advanced and very comfortable and useful than NMR, IR, and mass spectrometry. Explanation. Analysis of the diffraction data obtained from single crystals is the most important method of obtaining the structures of inorganic solids. It means when X-rays hit on a single crystal and then diffracted, those diffracted beams, when detected by a detector, and that diffraction pattern or the diffraction data obtained should be analyzed. When that data is analyzed, which is obtained from the single crystal, is actually the major or important method to obtain the structural information of inorganic solids. Provided a compound can be grown as a crystal of sufficient size and quality, the data provide definitive information about molecular and extended lattice structures. It means for this purpose, we should have to grow a crystal of sufficient size, as we have discussed in our previous lecture, lecture number 1, that the size of crystal is actually very important for a specific technique. The collection of diffraction data from a single crystal is normally carried out by using a 4-circle or area-detector diffractometer. A 4-circle diffractometer uses a scintillation detector to measure the diffracted X-ray beam intensity as a function of the angles shown in. This is the 4-circle diffractometer layout. The computer controls the location of the detector as the 4 angles are changed systematically. You can see on the screen in the center the sample is present and there are 4 different angles shown by the circle. You can see this at this image it is also the layout of a 4-circle diffractometer which is controlled by a computer. You can see at the center crystal is present and 4 circles are shown, which is maybe X, Y and Z axis and one at the center. An area detector diffractometer uses an image plate that is sensitive to X-rays and so can measure a large number of diffraction maxima simultaneously. Many new systems use this technology because the data can typically be collected in just a few hours. Analysis of the diffraction data from single crystal is formally a complex process involving the locations and intensities of many thousands of reflections. But with increasing advances in computation power, a skilled crystallographer can complete the structure determination of a small inorganic molecule in under an hour. A single crystal X-ray diffraction can be used to determine the structures of the vast majority of inorganic compounds when they can be obtained as crystals with dimensions of about 50 by 50 by 50 micrometer or larger. You can see this. This is the X-ray diffraction pattern obtained by a single crystal. The individual spots arise by diffraction of X-rays scattered from different planes of atoms within the crystal. You can see the light spots shown in a row actually show the atomic positions. The position for most atoms, including C, N, O, actually the carbon, nitrogen and oxygen, and metals, in most inorganic compounds can be determined with sufficient accuracy that bond lengths can be defined to within a fraction of a picometer. As an example, the sulfur to sulfur bond length in monoclinic sulfur has been reported as 204.7 less than or greater 0.3 picometer. A note on good or at least conventional practice: crystallographers still generally use the angstrom. Actually, 1 angstrom is equal to 10 power minus 10 meter, equal to 10 power minus 8 centimeter, and 1 degree angstrom also equals 10 power minus 2 picometer as a unit of the measurement. Actually, you can say the crystallographers still use the unit of measurement, which is the angstrom. This unit is convenient because bond lengths typically lie between 1 and 3 degree angstrom. The sulfur-sulfur bond length in monoclinic sulfur would be reported at 2.047 less than or greater than 0.003 degree angstrom. The positions of H atoms, hydrogen atoms, can be determined for inorganic compounds that contain only light atoms whose atomic number less than about 18 and the atomic number of argon is 18. You can say this is actually for the light atoms which are less than the argon. But there are locations in many inorganic compounds that also contain heavy atoms, such as the 4D and 5D series elements can be difficult or impossible. The problem lies with the small number of electrons on an H atom, hydrogen atom, just one, which is often reduced even further when hydrogen forms bond to other atoms. Other techniques such as neutron diffraction can often be applied to determine the positions of hydrogen in inorganic compounds. Molecular structures obtained by the analysis of single-crystal X-ray diffraction data are often represented in ORTEP diagrams. ORTEP is actually Oak Ridge Thermal Ellipsoid Program. In an ORTEP diagram, an ellipsoid is used to represent the volume within which the atomic nucleus most probably lies, taking into account its thermal motion. The size of the ellipsoid increases with temperature, and as a result, so does the imprecision of the bond lengths extracted from the data. Actually, we have discussed by increase in temperature, the vibrations or oscillations of the atoms present in a crystal is increased. If the oscillation or vibrations are increased, and the size of the ellipsoid will be increased, and if the ellipsoid size is increased, one would be confused about the lengths of bonds extracted from the data. Actually, that data is at higher temperature, and at higher temperature the bond lengths are not actually in their position. The advantage: analysis of the diffraction pattern obtained from single crystals allows the full determination of the structure. Ellipsoid, a three-dimensional closed geometric shape, all planar sections of which are ellipses or circles. An ellipsoid has three independent axes and is usually specified by the lengths A, B, and C of the three semi-axes. If an ellipsoid is made by rotating an ellipse about one of its axes, then two axes of the ellipsoid are the same, and it is called an ellipsoid of revolution or spheroid. You can say that you have ellipsoid and spheroid if all the axes, which are three, are independent of each other. They are ellipsoid. If two axes are same and one is different from the two, that will not be ellipsoid. That is actually spheroid. If the lengths of all three of its axes are the same, it is a sphere. So, you have three different terms: ellipsoid, spheroid, and sphere. In ellipsoid, you have all the three axes independently, and in spheroid, you have two axes same, and in the sphere, you have all the three axes same. You can see at the diagram there are three axes x, y, and z, and you can also see the three different circles. And if you have two axes same, one different, it will be spheroid. If you have all the three axes same, it will be sphere, and no one of the axes is same, that will be the ellipsoid. Procedure. The technique of single crystal X-ray crystallography has three basic steps. The first step and often most difficult step is to obtain an adequate crystal of the material under study. We have discussed we should have to obtain a crystal of sufficient size under examination. If its size is very small, then we will not be able to get internal structural information of the crystal. The crystal should be sufficiently large, typically larger than 0.1 millimeter in all dimensions. The most important thing, it should be pure in composition and should have regular in structure, with no significant internal imperfections such as cracks or twinning. Second step. In the second step, the crystal is placed in an intense beam of X-rays, usually of a single wavelength, producing the regular pattern of reflections. As the crystal is gradually rotated, previous reflections disappear and new ones appear. The intensity of every spot is recorded at every orientation of the crystal. In the second step, the monochromatic intense X-rays are irradiated on a sample, which is a single crystal, and these X-rays, when hit the sample, are diffracted, and these diffracted beams of X-rays from the single crystal are detected by detector, and we can get the diffraction pattern and can study the internal structure or information about the structure. And this crystal actually should be rotated in different sides. If the crystal is placed on a specific side and we hit the X-rays and get the reflections, when the crystal will be rotated, the diffractions or reflections we are seeing will be disappeared, and new side diffraction or reflections will be appeared. Multiple data sets may have to be collected, with each set covering slightly more than a full rotation of the crystal and typically containing tens of thousands of reflections. And third, the last step. In the third step, these data are combined computationally by a computer with complementary chemical information to produce and refine a model of the arrangement of atoms within the crystal. The final refined model of the atomic arrangement, now called a crystal structure, is usually stored in a public database. Thank you.